Welcome, guys, to our first ever episode of Let's Talk Bio podcast. Today, our topic will be about viruses.
Can someone please tell me what's a virus?
A virus is a small infectious particle that can reproduce only by infecting a host cell.
طب wait الحين I'm شوي كده confused. Is their structure different than cells, like living cells وكده, or like anything, but ولا they're like similar in like any way, shape or form?
Yeah, they're different. Living cells have organelles, but viruses have a different structure, and they have three main components.
The four main common features of a virus are the surface proteins, the capsid, the envelope, and the genome.
What's the purpose of the envelope? And do all viruses have an envelope?
No, a capsid reminds me of a soda for some reason.
An envelope is a part that surrounds the entire capsid. It contains proteins that help bind the infectious particles with the host cells.
What does the
So are they found in all viruses?
They are mainly found in animal viruses.
So not all viruses.
Only
So
animals.
it's a main structure, but it's not found in all viruses.
Yes.
What does the capsid do?
The capsid is a protein shell that is made up of many proteins. They join to make units called capsomes. They are encoded with the virus genome that provides the information to the host cell to reproduce.
So basically, the capsid contains the genetic material of the virus, which is very important in reproduction.
Yeah.
طب الحين the genetic material, is it يعني زينا كذا DNA ولا do they also like يعني have RNA ولا both ولا كيف يعني؟
It can be both. It can either be RNA or DNA. They can never be present at the same time.
Yeah, and there's a type of virus called retroviruses. They have RNA genome. Yeah, and 70% of viruses have an RNA genetic material.
طب does the يعني like do viruses that have DNA are different from viruses that have RNA? Like do they have like a different main thing, like something main that's different?
What do you mean? Like what?
يعني like, do you know what I mean?
I think, yeah, but retroviruses have reverse transcriptase, which is an enzyme that makes the transcription process opposite. يعني instead of the DNA becoming RNA or transcripted into RNA is transcripted into DNA, and this makes it different. And actually, makes it different. And actually, since it's not a normal process, it may have high mutation rates and mistakes, and any mistake in the genetic material is called a mutation. So that's why they have high mutation rates.
Wait, I remember, Noor, you mentioned that they need a host cell to reproduce.
Yes.
How do they reproduce?
And what's a host cell? To start, what's a host
A
cell?
host cell is so first of all, a virus needs a host cell to reproduce. It needs a human being, anything, an animal, anything to reproduce in. It joins a host cell and uses its machinery and metabolism of the host cell to make copies and produces. It also can. And I have a question for you guys. How does it recode the virus? How does it recode the
Genetic material?
Yeah.
the host cell? How does it recode genetic material of the host cell?
It injects its DNA and forces the host cell to change the material. it can either be integrated within the host cell's genetic material or it can be separate.
طيب وبعد guys for شو اسمه for the mechanisms of infection for bacteria بس like there are two cycles, lysogenic or lytic cycle.
So let's start off with the lysogenic since it's the beginnings. You can
Lysogenic basically leads to
Yeah.
lytic cycle.
So we can explain the lysogenic first and then after the lysogenic, the lytic.
Okay, so there are five main steps basically of like the lysogenic cycle when the when the bacteriophage attacks or infects the bacteria. First, it starts off by attachment. I think it's like pretty obvious by its name. So there are proteins like in the tail of the phage. There are some proteins. These proteins will bind to a specific receptor. On the surface of the bacterium. Then entry. So then the phage will inject its genome into the bacterium. Then integration will happen. Integration is when the phage DNA recombines with the bacterial chromosome and forms a prophage, which is integrated into the chromosome. So a prophage is like a main thing in lysogenic cycle. It's when the phage DNA is like integrated, it's like any with the bacterium's DNA.
And the prophage is not active, so it doesn't really affect the cell.
Yeah, it's like it doesn't affect it till later, till something triggers it.
Yeah.
So what will happen is the cell will like continue like proceeding and like living, growing and dividing. So when the cell divides, the prophage will be included within the bacterium.
It will be divided
DNA.
within the...
Yeah, it will be like copied and divided and everything. Just basically
It's part of this host cell's material.
exactly. But then eventually when something triggers the prophage, which will cause it to pop back out of the chromosome, which means like it will
Be separated from the host cell.
separate, but then it will
It will
it
be
will-
active.
and then it will enter the lytic cycle.
I have a
which will
question.
yeah.
What are those certain conditions that the prophage needs to undergo so it can start being active?
For example,
It can be stress, UV light, UV radiation.
And chemicals, and there is some.
also low immunity.
Yeah.
So these are just some of the things.
And actually, an interesting fact: when you're on your period, your immune system will be low, so that can be كذا يعني.
it will cause the prophage to
No.
like some parts of the body يعني the low immunity will when you have low immunity it يعني
Viruses target low immunity.
Yeah.
If you have low immunity, you're more vulnerable to a virus. A virus is most likely to affect you and really be the strongest while you're at your lowest.
yeah so
Yeah.
it will like
So you'll be prone to viruses.
yeah or lack of food will also cause
Lack of nutrition, lack of sleep, lack of...
So now I can explain the lytic cycle. The lytic cycle is a faster process, and it causes the cell to lyse. And the lysogenic cycle doesn't cause the cell to lyse. It doesn't affect the cell. So first, the virus will inject. The first step is attachment. Proteins found in the tail of the virus will bind to a specific receptor on the host cell. And the surface proteins in the virus are the key to the lock on the host cell. So the virus will enter the host cell by tricking the host cell. And then it will inject its genetic material into the host cell, and then the cell begins to make messenger RNA from the viral genes, or the viral messenger RNA is translated into viral proteins that will chop up the cell's DNA. Controlled by the viral genes, the host cell's metabolic system makes copies of viral nucleic acid. So we can see that the replication process is happening now, and the virus has created its nucleic acid.
So
The host cell, what? Do you have a question? The host cell's metabolic system also makes copies of capsid proteins. So now the virus has copied its genetic material and its proteins, and we will enter the assembly step. So the virus, the genetic material will be stuffed into the capsids to create the new viruses, and then the accumulation of the virus will cause the cell to lyse.
What does it mean to lyse?
Lyse means that the phage expresses genes for proteins that poke holes in the plasma membrane and cell wall. The holes let water flow in, making the cell expand and burst like an overfilled water balloon. And then after this, since it will explode, the virus will go on and infect other cells. Dana, can you tell me basically how it...
Okay, so basically, from what I understood from your explanation, lytic cycle, it's when a virus attacks its host. Correct me if I'm wrong, okay? It's when a virus attacks its host cell and it uses the cell's resources to make lots of new phages, but the cell won't lyse. Like it needs something to
هذا
trigger
the
it.
lytic ولا the lytic?
It
you
will.
are wrong. The lytic cycle, it causes the cell to lyse, to burst, to die in the process. In the lysogenic cycle, it allows a phage to reproduce without killing its host. When the phage DNA is inside the cell, it is not immediately copied or expressed to make proteins.
So in the lysogenic cycle,
It's basically a delayed process of the lytic cycle.
and the virus is in a dormant phase, which means that it's not active and it's not doing anything.
So how many types of viruses do we have?
We have many types.
But three main kinds. We have the animal viruses, plant viruses, and bacteria viruses.
What affects us humans?
The animal viruses.
The human viruses and the mutation of animal viruses can affect human cells.
Because you can get viruses from eating meat, because the animal,
Is it salmonella?
yeah,
Oh,
And guys, can I add something? I think that the lysogenic cycle really reminded me of like a prison jail. It's like when a prisoner is being held imprisoned إن واحد محبوس وبعدين خلاص طلعوه لأنه خلاص انتهى وقته.
I have a fun fact, guys. Did you know that viruses can be cooked up from scratch?
cooked up.
Yeah.
صدق؟
Yeah.
How can they be cooked up?
So what I mean by cooked up is not like actual cooking, but it's like cooked up like يعني basically they can be made from scratch in the lab. So they are so simple. The viruses, they are so simple. So it's possible to assemble one from scratch. you just need a few components, which is purified.
They put the virus in a pot and cook it in.
That would be fun though. But what they actually do is like they take purified protein and purified nucleic genome, and then they mix them in water under the right conditions for salt, acidity, and temperature. In time, those components will spontaneously organize into infectious virus particles.
Actually, so interesting. I love it.
Yeah, that's so interesting. I didn't know it was that of a specific process.
Also, Bahad, another fun fact which I found like so amazing and fascinating, that viruses evolve faster than any other living organism, because within hours thousands of copies can be made from a single virus. Since viruses cycle through multiple generations so quickly, they end up making frequent mistakes, and they live on their mistakes when copying the genetic information. They can rapidly change their form through these mutations. That's what I found from the UCLA Chemistry and Biochemistry website.
It actually reminded me of the cold virus. It's a shapeshifter. It has about 99 structures. That's why we can't find a vaccine to treat the virus. So yeah.
To be honest, viruses never fail to fascinate and amaze me. SubhanAllah.
And that's all for today's episode. Stay tuned for the next episode. We'll have a special guest coming over, or a special speaker. Don't forget to like, share,
and subscribe to our channel.
Let's talk bio
Podcast.
Bye.
Bye.